Okay, PowerPoint. You can see. Okay, so today we're going to start with chapter 1 and 2. I'm not covering the whole chapter because we don't have time, but I'll focus on the most important stuff for your exam and obviously for the future of this course. Okay, so I was thinking how can I start this course, right? And I think the best way to do it is to actually start by what most people understand as genetics. Okay, so the concept of like genetic inheritance, right? You get genes from your parents and whatever, right? Like some people are also breeders. It's been a long time that they've been breeders, and so they know that if they take this race of, let's say, whatever animal and then mix it with this one, they get a different animal. The whole horse, donkey, mule thing. Does anyone know what that little dog there is? Nope, you're close. Anybody? Come on, man. It's the cutest dog ever. It's a Pomshy, okay? It's called a Pomshy. So it's a mix between Pomeranian and Chihuahua, okay? So this is what I mean by the concept of like genetic inheritance. So you know that you get traits from your parents. The thing is, before, people thought that when you mix, like, let's say, two things, they will blend in, right? So if you have, like, for example, a blue flower and I don't know, white flower, then you mix them, then it becomes lighter blue. But that's not really how it is. It's a bit more complex than that. And this is where the field kind of started, right? And so the concept of genetic inheritance is used by, like, people that take care of, like, crops and whatever. And they mix and match different species to obtain better species, right? So for example, just my grandfather is a farmer, and he knew all this without knowing the genetics, right? So he was making like better crops each time, right? Okay? Whoa, whoa, what is happening? Press one button, you lose the whole thing. Okay. Where was I? Okay. So humans have 20,000 genes, okay? And these 20,000 genes make up each one of you, right? So only these 20,000 genes make, so the difference in these genes make you guys, right? And they're important, these genes, because they code proteins that are important for metabolism, structure, whatever, okay? Their role is actually almost not understood, okay? There's only a few of them that we understand. The rest we have no clue what the hell they do, okay? And some of them have like 20, 30 functions, okay? So what we know, though, is that we're able to affect the characteristics of crops, right, for, let's say, agriculture, to improve them, right? For example, genetically modified organisms. Usually we modify some sort of gene to make the fruit bigger, more colorful, whatever. And we can do this also on animals, technically on humans, although it's illegal and unethical for now, but there will be a point in the future where we'll be able to actually do that, okay? A good example of this is so this is part of your book. This is like a rice strain that when it gets submerged in water dies. Well, they just modify a gene called sub1, which comes from submerge, and then suddenly it's able to survive underwater, okay? And this gene affects many processes that I'm not going to get into because it's not really telling us much for this course. But if you want to read the details of this as an example, it's in the book, okay? One important thing, though, is that the genes do not only contribute to how we are right now, okay? There's also the environment. The environment has an effect on the genes, and this is called epigenetics, which we'll cover later on in the course. But it's basically what the environment is able to do to the genes, so it can activate certain genes or deactivate them, okay? So genetics, okay? Genetics, the way it started is that it started by being studied for genes that were like kind of clear to see, right? Short, tall, different colors, different size, whatever, okay? And the first genes that were described were inherited in a 3-to-1 pattern, and we'll get into the detail of what 3-to-1 pattern means in a few seconds, or 1-to-1, okay? These So one thing that is important to know is that to study genetics, you kind of need to study the population early on. Now it's a bit different, but you need to observe kind of some sort of phenotype, some sort of like change in the population, and then you know that these people have a different mutation compared to these guys, let's say, okay? So we're going to do a test just to give you an example of this. If you guys take your right hand and do this, and then see if your finger is actually straight or a little bit curved, okay? So that's curved, that's curved. Okay, so now what you're going to do is that I want to know how many of you have a straight finger. Lift up your hands. So guys, look around so you see what I mean. Most of the class has a straight finger, right? Now the other one's the curved. Go ahead. So what is this telling you? Does this look like a 3-to-1 or 1-to-1? It seems to be 1-to-1, right? Right? Well, this pattern here is basically telling you that it's only one gene affecting this curve here, okay? And you can see it in the population for many things, like simple traits, right? Eye color, for example, is a more complex trait. It contains many genes involved in it, so you can't really do this test, right? But like the hitchhiker thumb, you can definitely do that, okay? Another example is, for example, the celiac disease. Does anyone know what celiac disease is? Okay, well, I have it, okay? And the celiac disease is I'll explain this a bit later. It's a recessive disease. So this means that the general population does not have it, but 25% approximately does have it because the person has the two recessive alleles, which I'll get into in a second. So the two genes are mutated in the person, okay? Okay, so our Newton here. So the birth of genetics. The genetics actually started with a little bit earlier to Gregor Mendel, but it was not clear really what was happening. We knew that parents have offspring that look kind of like them, but we were not sure from where it came, right? So there's this guy called Gregor Mendel that was playing with pea plants, okay? So we have Newton with his apple, this guy with pea plants. If you want to succeed, choose a fruit and go for it, okay? What did I want to say? So he was playing with pea plants and breeding them together, and then he found that there is a pattern that was happening. And then he was able to conclude from it that genes behave like particles, and they do not blend, okay? It's not 100% true because now with the science that we have, we know that some of them do blend. So for his time, this is true, okay? And that there's always one allele dominant to the other. So an allele is a variation of a gene, okay? So it could be, for example, a gene produces a protein, right? And that protein, I don't know, it's an enzyme that does some sort of reaction, substrate product, okay? So what can happen is that you could have an allele that is even better than the wild type. So this is a mutant allele, so that with less substrate, so it's faster in converting this substrate to the product, okay? I'm just talking like in general. We'll get into the specifics of this, so just to give you an example, okay? You can also have an allele that loses the function of the enzyme, and so it cannot convert the substrate anymore. So this is a different type of allele, okay? What it means here by dominant to the other, it means that one allele is shown and the other is hidden, okay? So there's dominant, recessive, okay? And then what happened? So this guy published this paper, the father of genetics, as they call him, in 1866, and this paper basically transformed the whole field, okay? Since then, there were a few people that played with genetics, for example, Morgan, who found that genes are located in chromosomes, and then let's forget about Fisher because it's too complicated to explain for now. Beetle, who found that there's one gene, one protein, so each gene produces a protein. James Watson and Francis Crick, did you guys hear about those? Probably in CJ Bio, right? Okay. Well, these guys found out the structure of the DNA, the 3D structure, okay? And then I skipped a few steps. You go to 2001. That's when it was the first time that we published the genome of the human. So now we know what the genome of the human is made up of, and then we can start studying it in a much better way, okay? Lastly, in 2012, there was this new system that was created called the CRISPR-Cas9 system. They got a Nobel Prize for it in 2019. I should have put their pictures. And this system is amazing, okay, because now it allows you to cut things in the DNA and then insert exactly what you want, okay? As an example from my research, okay, I have a gene, the estrogen-related receptor, which causes cancer, okay? And then there's mutation, so I mutate it by just changing one of the nucleotides, and then I reinsert it into mice, and now the mice suddenly do not produce these tumors anymore, okay? So this is like some crazy stuff, okay? Instead, we're going to talk about our buddy James Watson, okay? I know you guys like a little bit of drama, so let's talk about this guy, this cool scientist that actually turned really bad, okay? So he won the Nobel Prize in 1962 for his discovery about the structure of the DNA. It was shared by James Watson and Francis Crick. I meant Watkins or whatever his name is, okay? And then suddenly this guy decides to go and claim that intelligence is determined by race, genetically speaking. Obviously, obviously you don't do that because even though we're in a free continent, let's say, and you can speak and whatsoever, right? People are kind of scared of this type of argument, right? So there's a way to show this scientifically speaking, but the community doesn't even want to try, okay? And this guy lost his Nobel Prize, his Cold Spring Harbor like title, and like the whole thing. Now he's like basically useless, okay? If you think about it a little bit, okay? This guy has been doing research for what, 60 years, right? He got to that thinking somehow, right? The problem is that the society did not give him the opportunity to kind of, they just cut him straight, like you're not politically correct, which obviously I'm not saying that race affects intelligence, I'm just saying like, check it out as scientists, right? So anyways, this guy lost his title, right? And now he's basically not doing anything. I think he's like 100 years old anyways. You can read this on the BBC website, but you know, super interesting source there for scientists. But his stuff is everywhere, and there is actually a field in genetics called eugenics, okay, which is even worse than what this guy is doing. It's called racial improvements, okay. So using genetics to modify the human to improve it, okay. These are like things I shouldn't be talking about because I might make a mistake and end up like him, but I'm not saying anything, okay? I'm just giving facts. So, okay, enough drama. Let's get back to the course. So Mendel's experiments, okay? We're gonna talk about his experiments, which will be very important for your exam, your tutorials, next classes, the whole, from now till like 3 years later, okay? So what he did, he was chilling in his monastery or whatever, and he was playing with pea, okay? He cross-pollinated, very important term, cross-pollinated, okay? Two pure lines together, and then he studied the inheritance of 7 traits. A trait is something like color, I don't know, shape, size, whatever, okay? Like the seeds, for example, the color of the seeds. So the 7 are right here on the right. Yeah, the right, okay. He also used another method called selfing. So cross-pollination is when you transfer pollen from one stigma to the anthers of another plant, a different plant. So this is why cross, right? Selfing is when you transfer the pollen from one stigma to the anther of the same plant, okay? And these two are important crosses because they will be used to solve, like later on, you will be asked probably in the exam and in the tutorials, XYZ did a cross-pollination between this and that, and then he selfed it, so, right? So understand these two, what you're doing with who, okay? I stole this slide. I borrowed it from a professor from here, but, and he drew this like, I don't know, a long time ago, beautiful flowers, okay? So this is just to explain a little bit the concept. Let me move this thing here. This is just to explain the concept of Mendel's experiment. So what he took pure lines of these peas, and then cross them together. So that's why there's an X there. And then the first, so these are the parents, the first line. Actually, you know what? Start using some cool stuff here. Where's the pen? Wow. Okay. Oh my God. Okay, I'm gonna buy a pen for this, so it's gonna be easier, okay? But basically the first line there is the parents, and then when he crossed them, he observed that the offspring are all blue also, okay? This is called the first filial generation, so F1 generation, okay? And then he crossed those offsprings together and still found that the F2 generation, which is like the group of like 8 at the bottom, all blue also. So this is what it means to be a pure breeding line. Okay, no matter how many times you breed it with itself or with another, it always gives you the same line. Okay. 1 second. Then he took another line that was that had flowers of white color. Okay, and did the same thing, and you can see that it gives you offspring of the same thing. Then he had the genius idea of breeding the white one with the blue one. Okay, and when he did that, so these are two pure lines. When he did that, he found that the F1 is still blue. And then he was like, what the hell is happening? Do you guys have an idea what's happening? Does one seem more dominant than the other? The blue one, right? Okay. This guy did not give up, so he continued breeding. So he took the first parents that were pure, bred them, found that they're all blue, and then took the offsprings of that, the F1, and then bred them together or crossed them between each other. And then he found that the F2 had a mix of blue and white, okay? And there's 3 times more blue than white, okay? We'll see why in a second. But basically, the white trait was hidden in the F1, okay? So he did this, I think it was 8,000 times, but the slides that I took are saying 1,000, but it's 8,000 times. And he found, let's say 1,000, and he found that 1/4 is white and 3/4 is blue, okay? Does this make sense up to now? Anyone lost? No? Okay. I hope you're ready, because now the serious stuff starts, okay? Let me see 1 second. I'm going to put this here. Okay. So, where's the other slide? There you go. You guys are aware of the idea of gene and alleles, right? Does that make sense? Anybody need an explanation of what an allele is? No? Okay. So what happens is that his idea was that, okay, the pure lines, so the pea seeds are diploid, okay? Let's start by that. So that means they have 2 copies of each gene, right? So he said, he labeled them like randomly like this. He said that the blue one is big P, and that's just 2 copies of the same gene that give the blue color, okay? And then he was thinking the other one should be small p, which is giving the white color, okay? When he bred them together, he found only progeny of capital P, small p, okay? Why? I'll explain that in a second. But basically, he was thinking, okay, this means that you only need 1 big P to give the blue color, right? If you have both small p's or both mutant alleles of the gene, then you'll end up with a white colored flower. Does that make sense? Kinda? No? Okay. He then took the offsprings of those 2 parental lines and bred them or crossed them between each other. So you have capital P, small p, crossed with capital P, small p, and that gives you this ratio here. So you would have one of the offsprings will be small p, the other one is capital P, and then the 2 of them will be capital P, small p, okay? I'm going to show you what this means here at some point. Yeah, we're going to get back to these flowers in a second. But basically, what he understood from this is 8 things, okay? This is exam material. Every prof likes to put it. I'm no different. So learn these by heart and understand them, okay? Because some questions will probably be on this. So Mendel's first law, from all these experiments, Mendel's first law is the law of equal segregation of alleles. Okay, so when your parents, when you have, like, for example, sperm and egg that mix together, they each bring one copy of the genome, and then it becomes two copies when they form the zygote. Okay, a diploid organism. So that's rule number 2, and then me or another or anybody basically that's diploid, when they're producing these gametes, they will separate into equally, right? So each gamete will have one copy of the gene. Okay, we'll get into the meiosis, mitosis, all that stuff later on. I just don't want to mix you up like from the go. But basically, each time there's this oogenesis that happens, there's 4 eggs that are produced, 3 of them die, 1 of them stays, right? For male spermatogenesis, there's 4 of them that get produced, but they all stay there, okay? Inside, there's 1 copy of this gene, inside each one of them, right? And this because there's a duplication event and whatsoever, okay? We'll get into this a bit later. This part is kind of confusing for the students. So I'll cover it by itself. I don't want to mix too much stuff, okay? So this is what it means by the equal segregation of alleles, okay? Because you're taking one from the parent and one from parent 1, one from parent 2, and then you're making those diploid organisms. So big P, small p, or big P, small p, okay? So the first rule, so this law is divided into 8 postulates. The first one is that there's discrete units of biological inheritance that are called genes, okay? Second one is each diploid organism has 2 copies of each gene. Diploid, 2 copies. Haploid, 1 copy. Triploid, tetraploid. Tetraploid is 4, etc. Okay? Are we diploid, triploid, diploid? Makes sense? Okay. I have to figure out the level yet of the students, so maybe it's going to get harder. Don't answer everything, you know. Postulate number 3, genes can have 2 or more forms. So this is what I explained a little bit earlier. You can have the allele that gives you the blue color, or the allele that gives you the white color, and then you can have a purple one and a pink one and whatsoever. Okay. For now, we're just going to focus on 2 alleles, 1 gene, but later on we'll see that you can have a whole spectrum. Okay. Rule number 4: the allele is either dominant or recessive. For example, big P is dominant. If you have only 1 copy, then you get the blue color. If you have 2 copies, you still get the blue color. And if you have no big P, then you end up with white color. Okay? What else? What else? The dominant allele always gives the phenotype. The phenotype is how the organism looks. Okay? In the heterozygote. This is very important. So if it's a dominant allele, like the color of the petals of the flower, then the phenotype of the heterozygote will tell you which one is the dominant and which one is the recessive one. Okay. This I think I just explained. Number 4 is just what I just told you about the separation of gametes, right? To produce 1 gene in each gamete, 1 copy in each gamete, and then they fuse in the zygote to make a diploid for humans. For yeast, it's a bit different. We'll cover that a bit later on, but don't want to mix you up. Okay. What else? What else? So for the gametes, since for example right now I'm diploid, right? So I have a copy from mom, copy from dad. Then when there's a replication of DNA that happens, okay, you'll end up with 4 copies. And then when these separate into the gametes, you'll basically have 2 that are similar from father, 2 that are similar from mother, because of the replication event. Okay. What else? The segregation is random. Not 100%, but that was in his time, it was random. But basically, segregation of these genes between the gametes is random. It's not determined, okay? So you can have equal chance of having whichever version of the gene, okay? And then this is what I said again. During fertilization, you have 2 gametes that mate at random, that fuse at random, and then you could have 2 recessive or 2 dominant, or a heterozygous that has 1 big P, small p. Okay, make sense? Basic life biology. Okay. Here's a little diagram to explain what I just said. So let's say, for example, parent 1 has the one version of the gene. So he has 2 copies of it, big A, and let's say, for example, the other parent has a small a. Then they produce their gametes, which contain only 1 copy of that gene, and then they fuse to form a zygote. You can see how 2 capital A's with, oh, shit, look at this. It's fancy stuff, man, touchscreen. So you can see how if you mix those 2 gametes, you'll produce a heterozygous zygote with 1 gene from the father and 1 from the mother, okay? And it will be big A, small a. If then you self this with itself, okay, selfing, so that means you're basically crossing the same genotype with itself, so big A, small a with itself, then it's going to be divided in a different way. Or if, for example, you're also looking, okay, I'm mixing up 2 things here, but it's the same principle, okay? So if you are mixing these 2, then you'll end up with, wait, let me backtrack. So the zygote is heterozygous for the A gene. It has a capital A and a small a. When the eggs are produced from this zygote, what will you get? You'll get 1 big A and 1 small a, right? And then if you produce, the sperm produced from this is the same thing, right? It will get 1 A, a big A, and 1 small a, right? So then if you fuse these together, then there is kind of a distribution that happens. So the overall ratio for the progeny from these will be 1/4 big A, small a, 1/4 small a in the corner, bottom corner left, right, and then 2 heterozygous, okay? Do you understand how this is happening? Am I going too fast? Easy? No problem? Okay. Good, good. Yeah, there's a lot of A's here. I'm getting mixed up myself. So, so this is what we call a 3-to-1 ratio. Okay, and Mendel, our buddy Mendel, found the exact same thing by looking at all the traits for his plants. Okay, now when you're looking at the population, you're not going to get a perfect 3-to-1 ratio. Okay, because population is not perfect. You'll get like 2.96, which is 3, and then 1 on the other side. Okay. This is exam question again, because you might get something like this, and then you're going to be like, oh, but it's not 3. So if you were not listening, then you're going to think that it's something different. Okay. So he looked at all the 7 traits, and all the 7 traits are inherited in a 3-to-1 ratio. What this ratio means, it means that a trait is controlled by 1 gene, and that it has 2 alleles. That's it. No 3 alleles, no 4 alleles. 1 gene, 2 alleles. Okay. And then the abundant class is the dominant allele. In this case, for let's say the flowers, it's the big P. Okay, the blue. Question. Practice a little bit. So a researcher is studying 2 populations of mice. One of them has small d, and the other population where all the mice have big D, small d. In this scenario, the small d would be considered a? Anybody? Jeez, I have to up the level here. Okay. yeah, homozygous and homozygous recessive. That's true. And then the second part? No. Think about it. It has to be one of the answers, right? It is heterozygous, but which one? I heard it somewhere. C? Yeah, it's C. So it's a monohybrid, okay? Monohybrid means 1 gene, 2 alleles, okay? We'll get to dihybrid, and it's going to go crazy after that, okay? Let's see the time. Okay, not bad. Whiteboard, Punnett square. You think I can make this work? Let's try. Have you guys ever used this? No? Okay. I'm discovering too, so let's see. Well, I guess no whiteboard. Does anyone know how to use this? Come on, guys, man. No? Yeah, I wanted to try it, but I know how this thing works. We didn't have this in my time. This is a new classroom. I think it's been like here for a year or something. Okay. Yeah. This one here?
Yeah, that's where you get
Ooh, my man. Right. That's it. I like you, buddy. Okay, where's the eraser here? Okay, so let me think about an example for the Punnett square. Yeah.
Sorry, do you mind saying it monohybrid?
Yeah, one gene, two different alleles, hybrid. Okay. Yeah. What's the difference between heterozygous and monohybrid? So that's a very good question. Let me think. I think monohybrid talks about the cross, while heterozygous talks about the, no. Actually, I'm not sure. That's a very good point. Let me verify that, and I'll send you something about this. Okay. Look at that, putting me on the spot, huh? Not cool. Okay. Let's see. Let's take an example. Okay. Let's say the gene for celiac disease. Okay. We'll do that one. So the gene itself is called HLA. See, the prof teaches you something simple, big P, small p, and then he goes with the DQ2, okay? Human leukocyte oh my God, this thing. We got this. Human leukocyte antigen DQ2, okay? What this gene does is that it detects, so you in your body right now, you have your own proteins, and your immune system does not attack your proteins because it knows it's yours, okay? This is one protein that's important for that, okay? So when people have celiac disease, for example, they have mutations in this gene, okay? So if we call this gene, I don't know, let's call it C, okay? So C gene for, can you read my disease? Can you understand? Make sense? Okay. Do you see it there? Shit, this is cool. So C is the gene for celiac disease. Okay. Let's say, for example, for you to have the gene, for you to have the disease, you need one copy only of the gene to be sick. Okay. So if I were to do a cross between, let's say, mom, which is like this, and then let's say the father, which is like this, okay? So the first step, if you get a problem like this, what you need to do is separate the gametes, right? Because then they're going to fuse. So for the mom, what are you going to have? You're going to have big C here, small c here, right? And then if you make your little Punnett square, for the father, you'll have C, C, right? So then the gametes produced by this are what? They're going to be big C, small 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 c. Does that make sense? Oh, sorry. Yeah. I'm going to find it. I think it's this one here. Wait, we're getting there, guys. Actually, I'm giving you an example from that I know, which is, wait one second. Oh my God. Sorry guys, I'm getting used to this, and then it's gonna be, man, this is useless. Okay. How do I make this smaller? Press less. Wow. Okay, does the mom have it? So if I tell you that it's dominant, right? If I tell you that it's dominant, does the mom have it? Right? Does the dad have it? No. Okay, well, this is my mom, this is my dad. Okay, my mom has it too, right? So then my parents had 4 kids, actually. This is not even a joke. They had 4 kids, okay? So if you do the cross like this, mom, dad, how many of them have celiac disease? 2, and how many don't? 2. Well, my sister has celiac disease, the other 2 don't have it, okay? So this is actually like very clear in terms of inheritance, okay? Even more, my mother's brother has it, and his daughter has it, okay? Do we say this is a sex-linked trait or a autosomal trait? Is it linked to, sorry? Autosomal, right? Because if only like one of the 2 sexes had it, then you can say it's sex-linked, okay? Cool. So that's my family. All right, not bad, not bad. We're learning this stuff here. As the semester goes, it's going to be much more better. Do you see the slides? Okay. So this brings us to the next topic, the next type of cross, right? What did I want to say? So for just a quick review, so if we are crossing the homozygous, now the terms are important, homozygous dominant with the heterozygous, this is the only type that you'll have, okay? This is the only result you can have, because you'll have one big P crossed with one big P and one small p, right? Does that make sense? Because all the gametes from one parent are the same, okay? So you'll end up with a 1-to-1 genotypic ratio. Does that make sense? Yeah? Okay. If you are crossing 2 heterozygous, then the genotypic ratio will change. You will have 1 to 2 to 1. What that means is that you'll have one homozygous dominant, the big P. You'll have 2 heterozygous, which will be big P, small p, and 1 homozygous recessive, which will be small p. Okay? Now it's important to differentiate phenotypic ratio with genotypic ratio. Okay? Genotypic ratio, it's the ratio of alleles in your or genotype inside your plants. Phenotypic ratio, it's the appearance ratio. You can see here that both the homozygous dominant and the heterozygous have the blue color. So that's why the phenotypic ratio is 3 to 1. Does that make sense? Okay. There's another type of cross that is very important to learn, which is the test cross. So let's say, for example, I give you, exam question, if I give you a plant and I tell you find out the genotype of this plant, the only way for you to figure out its genotype is by crossing it with one of the pure lines, right? Because this way what's going to happen is that you'll see the genotypic ratios change, and then you'll understand if it's big P being crossed with the other one or the heterozygous. Does that make sense? Okay. So the test cross. For example, I give you the blue one there and I tell you figure it out, what is the genotype for that one, and you have no idea. You will cross it with the other one, and then I tell you the phenotypic ratio is 1-1. That means I get equal numbers of white and equal numbers of blue. So from there, if you look at the Punnett square, you'll realize that actually the parent is homozygous recessive, okay? Just like this here that we just showed, okay? If, for example, it was the parent was homozygous dominant, but we didn't know that, and then I ask you to test cross it, and I give you the Punnett square as a result, what would be the genotypic ratio? We're going to see that right here, I think. Did I put it? Wait. So if you do big P with small p, what do you get? Sorry? 3 to 1. Big P, small p. So that's PP. No. You will get all the same, which will be heterozygous, right? So let's do that one, actually. Another reason to use this guy here. Okay, cool. Okay, so let me ask the question again. So I give you this blue flower here, okay, and cross it with, and then I give you, for example, a Punnett square that looks exactly like this. So blue blue. And I ask you to figure out its genotype. How do you do that? You test cross it with one of the pure lines, because the pure line will show you the hidden traits, okay? So you cross it with, let's say, P, right? The only way for you to have blue, blue is if this guy here is big. Yeah, I chose the worst letter ever. So this is capital, this is capital P, this is small p, okay? So if you do, okay, you know what? Let's do big P like this. Man, this is super sensitive. And then let's make a little line here. This is small p, okay? Okay. So this is the only way that we would have the blue phenotype everywhere, right? Because if you mix this guy with this guy, you'll end up with blue, right? Because this is a dominant trait. Same thing here, same thing here, and same thing here. Okay? Make sense? A little bit mixed up, but this is something that is asked a lot on exams. Okay? So, yeah? Sorry? Oh, yeah, speaking of that. So we are not covering yet sex-linked traits. We're going to do this next lecture. I don't want to mix you up too much, okay? So this is, for now, just for autosomal, okay? Sex-linked is a bit different because it's linked to the X chromosome and Y chromosome and then things like this, okay? Any questions? No? Maybe? Okay, let's get back to this guy here. Okay, so this is that. Okay, some important ratios to remember, okay? This is basically what I just said. If you cross two heterozygous together, you'll end up with a phenotypic ratio of 1 to 3, so 3 blue, 1 white. And then the genotypic will be 1 to 2 to 1, which basically means 1 homozygous dominant, 1 homozygous recessive, and 2 heterozygous for the genotype, okay? If you're test crossing a heterozygous with a pure line, so the small a, then you'll end up with a 1-to-1 phenotypic ratio, and 1-to-1 genotypic ratio. Namely, half of them will be blue, half of them will be white, and then half of them will be big A, small a, and half of them will be small a. Okay? We just did the third cross, which is similar to this one, but gives you, all of them blue. Okay? Make sense? Okay, next one. Pedigree. So I was talking about my family and how they got celiac disease and all that. So the way we study this in genetics, well, the way they used to study it in genetics, but we're still learning it, is through pedigree analysis. So pedigree is a kind of a schema that's used to show how a gene is, or how a mutation is carried on through the family, okay? Circles in the pedigree is the female, square is the male, and then if the square or circle is filled out, that means the person is affected by that mutation or that disease, has the disease, okay? And then there's like 8 other different types of squares and all that, so we're not going to get into those because it's going to get too complicated. For now, just focus on the square, circle, filled or not filled, okay? So here, did you guys ever see pedigrees before? You did. Okay, so this is just a little review for you guys. Perfect. So the way it's drawn here, the parents are the first line there, and then when they have offsprings or the siblings, offspring, sorry, they're shown on the second line. Okay, male is square, female is circle, and then the dark circle means that the person has the disease. Now you could be asked, for example, to determine if this is a recessive allele or a dominant allele. And the way you know this is by trying to play with those genotypes and whatsoever. You can see here that if it was dominant, that means one of the parents would have it, right? There's no way that all of them don't have it except this person. So this is clearly a recessive. And if you look at the proportions, there's more people that don't have it than people that have it. So a recessive allele usually is the rarest form of the gene, okay? there's only one thing. So it's not gray and white, black and white. Okay. Reality in nature, it's a bit more complex than that, because even if the parents don't have it, you can develop it. Okay. For example, allergies. Your parents don't have it, but then suddenly you become allergic to things. Okay. Or for example, well, for my case, for celiac disease, my mom had it, but she developed it after me, right? So I don't know if that makes sense. So sometimes what happens is that the mutation actually happens spontaneously. Okay, this is a less likely possibility, but it could be also, this diagram could also be explained like this. Okay, good. So then this daughter decided to go get kids. Okay, so then the offsprings of this daughter with a non-affected male gave this result. So one child that has the disease and the other that does not have it. Based on this information, do you think that it's recessive or dominant? Sorry? Yeah, why? Go, Pedro. Yeah. Yeah. The disease? Okay, so just to repeat it, yeah. Yeah. So, okay, I see you did your reading there. Okay, so let me just re-explain this. So the way you play with these pedigrees, you can actually just put the alleles and try, okay? You can assume that it's recessive, try it. If it doesn't work, assume that it's dominant and try it, okay? But if you can take a look at this, none of the parents has it, and then suddenly it appears in one of the children, but not the others, okay? So, and the majority don't have it. So this looks already like it's recessive, okay? So if we assume that it's small a for that daughter that has the disease, and then gets children with another, with a male that doesn't have the disease, and then suddenly the children, one has it and one doesn't have it, you can even deduce that the other parent is heterozygous, okay? And if you try it and you do the Punnett square, you'll end up with a recessive trait. Okay, so try this. You'll be practicing all of this during the tutorial. I can't, go through all of them, but the TAs will give you problems like this, and then you'll be solving them. Okay, and then they will reappear in the exam and final exam, and then you won't see it again. Okay. Next, pedigree analysis example. Well, actually, I'm not going to do it on the whiteboard this time. I'm just going to let you guys try it, and then we'll go to the questions after that. There's 4 questions. We solve those. We go home. That's it. Okay? So in this diagram, you can see that there's 2 parents that don't have the disease, which is autosomal again. And then they have offsprings. 2 of them don't have the disease, and 1 of them has the disease. Okay? Now they will ask you, what's the genotype of I1? Are you guys able to determine the genotype? Yep. Were you going to say that? Okay. So the genotype is heterozygous. Can you explain why, please?
Because the argument that the disease must be recessive.
Okay. You're saying it's recessive because most of the children don't have it, right? And then 1 of them has it. Okay. So that does make sense. If you learn, let me go back. If you learn these, like, just print them in your head, you'll know exactly without even reading the question. Okay. But you'll practice this more. Okay. So the answer should be B. Good. A+, man. Let's go. Okay, what about the genotype of double I1? Or 2, 1. Yep. It,
Since it doesn't have the disease, so it's going to be either homozygous for the dominant allele, which is not safe, or it's heterozygous.
okay. Do you guys agree? Yeah? Speaking of which, do you like this type of question or no? Not really. Because I remember like in organic chemistry, the prof would give you like a 13 questions on the same synthesis, and then if you fail one, you fail the others. So I don't know if like I should incorporate those or not. Maybe I'll make it like 2 or 3 questions in a row, but not more than that, okay? You are right, buddy. You are right. This is good. One thing that I'm realizing is going to be very hard to edit out people, okay? So if you don't want to be on the recording, send me an email, and I will edit you out, okay? I think this is easier like this for me, because otherwise I'm going to spend like my whole time on video editing, and no, we're not going to do that, okay? Questions? Does everyone understand this? Yeah? Exact. So they're linked. So because you know that it's recessive, right? You just answered saying that it's recessive. Then that means the other 2 genotypes that are possible will give the healthy one, right? And then wait, what am I doing here? I mean, it could help. It could help, but it could. So you know what? We'll do it on the board. Why not? Okay. But then I can't show this. Okay, I'll draw it. No worries. We got this. Okay. So it was. Jeez. So it was like this, and then their kids were something like this, I think it was. And then the daughter had the disease, right? Okay. If it's a dominant trait, one of the parents has to have it, right? Because both parents don't have it, and one, what did this come? Huh? Yeah. Yeah, it doesn't, it's not really like, I'll still do it, but yeah. Yeah. When we're going to talk about like sex-linked traits, then yeah, the daughter, right? But in this case, not necessarily, but I'll still do it to keep it clear. So because both parents don't have the disease, it means it's not dominant, okay? It means it's recessive. And the other point that's telling us that it's recessive is the fact that most children are healthy except one of them, okay? So we can assume that this one is AA, okay? Small a, do you see it? Okay. This here now, so in a Punnett square, where you would have, so let's say, so this one here would be the one with the disease, okay? This one would be the small a, right? And then for you to get this kind of Punnett square, there's only one way to do it, okay? It's when both parents are heterozygous. Does this make sense? Okay. This is the diseased one. So what this leaves us with is 1 homozygous dominant and 2 heterozygous. 1 homozygous dominant, 2 heterozygous, okay? Now the question was asking, what is the genotype of this one here? Because it's healthy, it could be either this one right here or this one right here, or actually this one too, because these 2 are the same, okay? So this is how you answer the question. It could be either A or AA. Does that make sense? How can we figure out this, if this is big A, or a big A, small a, heterozygous or homozygous? Yeah? Yeah, but there's a specific cross that I showed you. Yeah?
We cross it with a
Pure. Okay, we don't do this with humans, but makes sense, right? So you would do a test cross. So if you were to cross it with a pure line, let's say small a, and you would get, so can I erase this? Yeah? Let's see. Oh my God, it's gonna be a Wait, whoa, whoa, whoa. I'm getting old here. Can you say that again? Nope. Wait. We're going to figure this out, guys. Let's try again. It's okay. Don't worry about it. When you guys are going to leave, I'm going to play with this. Okay. Let's go. I'm going to start saying bad stuff here. Okay. So if we cross it with a pure line here, this is called the test cross. And now the other one could be either big A, or small a, big A, small a. Let's start with the big A. So if you do big A, there's only one possibility, right? Which is like this. And then in the case, so then they would all be healthy. Does that make sense? If we cross it with this guy here, now we're going to erase this again. If it was like this, okay, so here, right. Jeez, it's horrible. Okay. So if it was heterozygous, then this genotype is going to change, right? Because even the phenotypic ratio will change. Because now what's going to happen is that you'll have half healthy and half that are diseased. Does that make sense? Okay. So this is how you solve this problem. Probably you will be asked, I think even in the tutorial, to figure out the genotype and the phenotype of a person like this. Okay? Questions? No? Okay. So let's get back to this. Let's get back to this. Next. So now the question is asking, what is the probability that if the two parents had another child, that he would not have the disease? Yeah? No. Look careful. 3/4, exact. Can you explain why, please? Sorry, it's what? Yeah. 3-1, exact. So then, exact. Okay. So, did you understand? No? Yeah. Okay. The only way for you to get, if it's recessive, the only way for you to get this is by having two heterozygous parents. Okay. And we know that the Punnett square for two heterozygous parents is 3/4 healthy and 1/4 not healthy. So now it's asking you, the fourth child, would he have the disease or not? What's the percentage of chance that the child would not have the disease? It's 3/4. Okay? Good. And last question. Not bad. Very good on timing on top of that. So what's the probability that the number 2 or II3 is heterozygous for the dominant and recessive alleles? Do you have an idea? Sorry? 2, 3? How come?
Because you know he's unaffected, so the chances having something increase. Unaffected, then you can either be homozygous dominant or heterozygous.
Yeah.
Heterozygous comes up twice, homozygous once, and overall that's 3, 2 of it.
That's it. Make sense? Yeah? Yes. Okay. So the probability, whiteboard. Let's go. My new best friend here. So the question is asking, what is the probability that this one is heterozygous for the dominant and recessive alleles? Okay? Heterozygous. So if we look at, so the possibility that that one, so it's healthy, right? So healthy means either A or AA. Make sense up to now? Okay. Now in a Punnett square where you're having two heterozygous parents, you'll have a ratio of 3 healthy, 1. Actually, you know what? I'll do it completely so you can get me right there. That's it. So AA. So now we need big A here. Just 1 second, guys. So this is parent 1, parent 2. No? Yo. Yeah. So this is parent 1, parent 2. So when you cross them, you'll end up with something like this. So now it's asking you what's the percentage probability that you'll have a heterozygous for the dominant and recessive allele. Dominant and recessive. So this here, right? This heterozygous, right? And because he's healthy, that leaves only 3 options, right? Either this, this, or that. So it's 2/3. Make sense? Good. So the healthy part is the tricky part of the question, kind of. Make sense? Questions? Okay. So that's it for today. Just a few things before you leave, because I know you're going to. That's it. Next week, the tutorials will start. Okay? On Monday, who has the first tutorial session on Monday? Okay, I'm going to be your TA. I'm replacing for your TA because she's on a conference. She's in North Pole, I think, or somewhere like that, studying some bacteria or something like that. So she'll be back the week after, okay? In the afternoon, the TA is there. On Wednesday, there's another TA that's going to replace. So this week, the tutorial session on Monday, 1:30 to 3:30, and the Wednesday one, the TA is not your TA. It's either me or another TA. The week after, you'll get with your real TA, okay? I'll send you some questions to do. You don't need to do them at home. You'll practice them in the tutorial, okay? Make sense? Take care, guys.